And explanations on how enzymes function as catalysts in biochemical system are based on the concept of an enzyme active site and enzyme substrate complex formation. Studies show that only a small portion of an enzyme molecule called the active site participates in the interaction with the substrate or substance during a reaction. The active site, as definition, is the relatively small portion of the enzyme's structure that is involved in the catalysis. So basically, active site is this small portion. The active site in an enzyme is a three-dimensional entity formed by groups that come from different parts of the protein chain. These groups are brought together by the folding and bending, in which that is your secondary and tertiary structure of the protein. The active site is usually a crevice-like location in the enzyme. Catalysts offer an alternative pathway with lower activation energy through which a reaction can occur. In enzyme controlled reactions, this alternative pathway involves the formation of an enzyme substrate complex as an intermediate species in the reaction. As defined, enzyme substrate complex is the intermediate reaction species that is formed when a substrate binds to the active site of an enzyme. Within the enzyme substrate complex, the substrate encounters more favorable reaction condition than if it is were free. To account for the highly specific way an enzyme recognizes a substrate and binds it to the active site, researchers have proposed several models. And the simplest of these models is the lock and key model, in which it is proposed by Emil Fischer. So the lock and key model is the active site in the enzyme has a fixed, rigid geometrical conformation. Only substrate with a complementary geometry can be accompanied at such a site. Much as a lock concept, only certain keys can enter the lock. That's why it's called lock and key model. Again, for the lock and key model, only with the similar geometrical shape with active site the substrate can bind into. The next model is the induced fit model by Daniel Koshland. So here, there are experimental evidence indicates that many enzymes have flexibility in their shapes. They are not rigid and static. There is constant change in their shape. The induced fit model is used for this type of situation. The induced fit model allows for small changes in the shape or geometry of the active site of an enzyme to accommodate a substrate. A good analogy is the changes that occur in the shape of a glove when a hand is inserted into it. The induced fit is a result of the enzyme flexibility. It adapts to accept the incoming substrate. Let's now proceed with the enzyme specificity. So basically, for the enzyme specifically, it is the extent on which an enzyme activity is restricted to a specific substrate. Absolute specificity is basically a reaction in which it says that there is an absolute substrate for an absolute enzyme. For example, we have here hydrogen peroxide. Hydrogen peroxide is the only substrate that can react into catalase. Next is for group specificity. So for the group specificity, it's still specific with a certain group. However, instead of a specific substrate, they are specific of a functional group. As long as they have the same functional group, they can accept it and react into. Next we have here is the linkage specificity, in which this is a particular type of a chemical bond. This is the most general of the common specificities. For the stereochemical specificity, so they are specific with one isomer only. So they are basically similar in the molecule, or the substrate is similar in the molecule. However, they as they are very specific with a stereochemical, they will only act on this specific type of isomer. Let's now proceed with the factors that affect the rate of activity of enzyme. So we have here the following factors: the temperature, the pH, concentration of substrate, and lastly the concentration of enzyme. To further understand, let's have here the graph. So for the temperature and pH, we have here is like a bell. Why? Because on its optimum temperature or maximum rate of the pH, in which, again, as I have told you, that enzymes are very specific. So therefore, for example, let's have here the enzyme amylase. Enzyme amylase is very specific with its temperature and pH, in which it can only live for a 37 degrees Celsius or a body temperature which is 35 to 37 degrees Celsius, while a 7 pH. If and when there is a slight change of the following, like for example, you will have a fever, amylase will now be degraded based on the change of the environment. For example, you have a fever and then that temperature will increase into, let's say, 38 degrees Celsius. The tendency for the enzyme amylase is that it will drop sharply. Therefore, in the graph, it will be decrease, or the number of substrate it can produce will be decreased. So since our amylase or our enzyme amylase will degrade the starch, if and when the patient is having a fever, tendency for that, the amylase will be degraded, and the tendency for that is that the starch the amylase that needs to make into a product will also be decreases. Similar also with the pH. For the substrate concentration, you have to remember that substrate concentration or the substrate is very independent with the number of enzyme. As long as there is an enzyme, the substrate will be continued to be a product. So for example, we have a 5 enzyme. So the 5 enzyme will also react with the 5 substrate. So what will happen here is that the enzyme will react to the 5 substrate, making a 5 product. However, if there is a tendency that the substrate concentration will increase, like for example, you still have the 5 enzyme, however, the substrate will become 10 substrate. So there is an increase of concentration of the substrate. So what will happen here is that the enzyme will only react to the 5 substrate. So the other 5 substrate have to wait for its time to be able to bind to your 5 enzyme. So it will only happen as long as all of the enzyme that is binded with your all of the substrate that is binded to your enzyme will be totally reacted with the enzyme. And then the other enzyme which make your substrate into a product will now or will can react now with the new or to the other substrate. That's why we have here a plateau, which is a graph for a substrate. Lastly is the enzyme concentration. For the enzyme concentration, again, enzyme concentration is very independent with your substrate, meaning as long as there is too much enzyme, there is also be a increasing reaction. Or for example, you have a 5 enzyme and 5 substrate. So basically there is a normal reaction. However, if we will increase the enzyme concentration, if for example instead of 5 enzyme you have the 10 enzyme and still 5 substrate. So what will happen here is that your enzyme will make the substrate or will react with the substrate faster comparing with the 5 enzyme only. So, as you can see in the graph, it is a increasing reaction. So that is for the factors affecting enzymatic activity. There are some enzymes in which they can actually live in a greater environment comparing with a normal enzyme. That is also true with microorganisms. So that's what we call extremophiles. From the word itself, extreme, live in a very they can live in a very extreme environment. Some example of which are the following. We have here acidophile, alkalifile, halophile, hyperthermophile, cryophile, and piezophile, in which acidophile can grow on extreme pH, in which that is 3.0 or below, and alkaliphiles, which they can grow on pH level which is very basic, that is 9.0 or above, halophiles that can exceed zero point 0.2 m sodium chloride needed for growth, and hyperthermophiles in which they can live in extreme temperature, cryophiles they can live in extreme drop of temperature, and piezophile in which they can live in a high hydrostatic pressure needed for growth. The best example for extremophile that can actually live in the human is your H. pylori, in which you can see them in the stomach ulcer. Helicobacter pylori, or also known as your H. pylori, is a bacteria that can actually function or that can actually grow in a very acidic environment of the stomach. So what will happen here is that they are the one who will make a hole in your stomach ulcer, and our main goal here is to make our stomach pH higher, because H. pylori can only live in extreme acidic environment. That's why one of the treatment for eliminating the H. pylori is the acid suppression or acid neutralization medication. Some of it is to kill the bacteria, in which they will use the antibiotics tetracycline, amoxicillin, metronidazole, clarithromycin, and levofloxacin.